All right, I'll be there in a minute. All right, we got to make this quick. I got to go and feed the kids some lunch. So here are the objectives. We will give an overview of protein synthesis. We will describe and model transcription. We will summarize a comparison between DNA and RNA. Here we go. Let's get back to the basics briefly. We have this thing called DNA. It's in the shape of a double helix, and that double helix is made up of subunits called nucleotides. And those nucleotides are made up of phosphates, sugars, and 4 different types of nitrogenous bases. This picture here is a nucleotide. This is the phosphate, this is the sugar, and this is one of 4 nitrogenous bases. So to the right, we see our backbone to DNA. On the left and on the right, that's what we call the backbone, alternating phosphates and sugars, and the rungs of a ladder, because it kind of looks like a ladder, the nitrogenous bases that complement each other. The C matches up with a G, the A with a T, the G with a C, and the T with an A. All right, so that gives us and reminds us of our DNA structure. Remember, we did something called DNA replication. In this DNA replication, what we did was we recognized just those nitrogenous bases because the phosphates and the sugars always go together. And we identified the DNA strands separating, unzipping with the use of an enzyme called helicase, and rebuilding the complementary strand. And it would look like this. And on this side, an A would match up with a T, T with an A, T with an A, C with a G, G with a C, G with a C. And we recognize that they made an identical copy of the original strand. But now the story gets a little bit richer. On the left here, we see a nuclear membrane, which means where I'm drawing in now is inside the nucleus. And this is that double-stranded of DNA, the double helix. But here's another strand, and it looks similar, but instead of T's, we have U's. And it's only single-stranded, and it can leave the nucleus. And this is a critical part of being able to make a protein because that DNA cannot make its way out of the nucleus to, if you remember, ribosomes end up building the proteins. But they need the instruction from the DNA. And it's this molecule that becomes our focus. But we need some definitions first. All right, protein synthesis, the process where the code found on genes, DNA, is used to build specific proteins. It utilizes 2 steps, transcription and translation. Transcription, a process where the code of DNA is converted into a new code, mRNA. Translation, a process where at the ribosome, that's not in the nucleus, at the ribosome, the code of mRNA is aligned with the code of tRNA, 3 letters at a time. The tRNA brings with it a specific amino acid, ultimately forming a chain of amino acids known as a protein. So we are going to need to know about that, but the rest of this video is going to focus on just transcription. And for that, we are going to bring in a video, and I'll play it, but I want you to take note in how it compares to DNA replication. So let me bring that into our screen. A little bit too large. There we go. There's a good fit. And let's play.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene.
All right, so they said a gene, but they didn't say what this coiled stuff is. It kind of looks like an old phone cord. If you zoomed in, you'd notice that it is the double helix. This is the DNA, and those are the factors that they just mentioned.
A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
Unzipping. You see, here's the DNA together, the two complementary strands, but over here they have been unzipped. They're re-zipping back together, but here they are unzipped.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
All right, so that animation was actually quite incredible. And let's focus on just a portion that they were speaking of. Highlighted in blue here are the two strands of DNA, unzipped by this substance called RNA polymerase. And it ends in ASE, so we know that that is an enzyme. So this enzyme is opening up the DNA strand, unzipping it to reveal the other nucleotides. Let's call them naked nucleotides so that new nucleotides can be added. In this case, we're going to be building a strand of RNA, in this case, mRNA. And the M stands for messenger. It is sending the message of DNA outside the nucleus. I want to show you this as well because it's more of a global picture. Inside the nucleus, once again, we have the double-stranded DNA. Built from that DNA is mRNA, which is leaving the nucleus and going to a ribosome. Now, these pictures are out of scale, but they are a good representation. All right, so now that we know about DNA, we reminded ourselves about the nucleotides. Let's see how the nucleotides for mRNA are built. So here's that same strand of DNA from before. Let's unzip it. And let's see what nucleotides would join if it were mRNA. So A would normally go with a T, but remember, T's are replaced with U's. So put a U here. A T would match up with an A, a G, a C, and a C. And what would then happen is these nucleotides would leave the nucleus together. The DNA would rezip, and this would go to the ribosome for the next stage called translation. Again, this is transcription. So that summarizes it, but I want to give you some notes, another way of visualizing the differences and similarities between DNA and RNA. So what do they have in common? Well, they're both organic. They're both made of nucleotides, which means that they have a sugar, a phosphate, and a nitrogenous base. Right? DNA is called deoxyribonucleic acid, the D, N, and the A, while RNA is called ribonucleic acid, the R and A. DNA is double-stranded, while RNA is single-stranded. DNA replicates itself. RNA needs to be built. The mRNA gets built from the DNA. DNA, the sugar is deoxyribose, while in mRNA, the sugar is ribose. Oh, and just like enzymes end in ASE, These are sugars. They end in OSE. And lastly, uses the nitrogenous base thymine, while RNA uses uracil. Otherwise, the other three bases are the same. All right, so that summarizes transcription. Next video will be about translation. All right, it's time for lunch.